Hi, this is Dr. Rosario. In this lecture video, we are going to be talking about prokaryotic cells, those cells that don't have a nucleus within the cell. So this includes our bacteria and our archaea. In this lecture, we're going to focus primarily on the different structural characteristics we see within our prokaryotic cells. So we're really going to focus mostly on bacterial cells. We're going to look at the different types of cell envelope we see in our bacteria, and what I'm talking about here is our gram-positive cells and our gram-negative cells. And then we're going to look at the different structures that we find inside of our bacterial cells, inside the cytoplasm. We're going to discuss some of the differences that we see between bacteria and archaea. We don't talk a lot about archaea in this class, so we want to touch on that just a bit in this lecture. And then finally, we're going to discuss some of the methods that we use to group and classify our prokaryotes. So before we start thinking about the specifics of our prokaryotic cells, we want to consider the characteristics that are common to all types of cells. So all cells consist of four main biological molecules. And we talked about these back in chapter 2, right? So you have your carbohydrates, right? Your sugars, your polysaccharides. We have our proteins. Remember, proteins are built from amino acids and they perform a lot of the work that occurs within a cell. Your lipids, right? We have phospholipids and steroids and your triglycerides. Those are all examples of types of lipids that we find in cells. And finally, nucleic acids. So remember, nucleic acids are used for information storage and information transfer inside cells. All cells use DNA, or deoxyribonucleic acid, to store genetic information. The way that the DNA is packaged inside the cell might vary a little bit between our prokaryotes and our eukaryotes, but both groups of cells use DNA to store genetic information and then to pass that genetic information down to their offspring. All cells have cytoplasm. So cytoplasm is like the jelly center of the cell. It's that fluid-like matrix that we find inside the cell. All cells have ribosomes. So ribosomes are the machines that are used by cells to build proteins. And finally, all cells have a plasma membrane, right? That separates the inside of the cell from the outside environment. So we know that there are two kinds of cells, right? There are prokaryotic cells and there are eukaryotic cells. Remember, prokaryotic means before the nucleus, and eukaryotic means true nucleus. So our prokaryotic cells do not have a nucleus, whereas our eukaryotic cells do. So because our prokaryotes do not have a nucleus, the DNA for those cells is just floating around freely in the cytoplasm of the cell. In contrast, your eukaryotes, they have their DNA stored inside this double membrane structure, right? This nucleus that is the biggest organelle we find inside of our eukaryotic cells usually. Your prokaryotic cells in comparison to eukaryotic cells are smaller and, quote, simpler cells. They're by no means simple, but they're less complex usually than our eukaryotic cells. And our eukaryotic cells tend to be larger and they tend to have more internal features to them. They tend to be more complex. They contain what we call membrane-bound organelles. So all prokaryotic organisms are single-celled, right? They're made up of a single cell. Our eukaryotic organisms, on the other hand, they can be single-celled or they can be multicellular. Bacteria and archaea are considered prokaryotes, right? But then your eukaryotes include plants, animals, fungi, and our protozoa or protists. Right? So that gives you a good comparison between the two groups. Prokaryotes are smaller, simpler. The big thing is they don't have that nucleus. All right, so let's take a look specifically at our bacterial cell. So this is an example of a bacterial cell, and you can see, yes, it is a relatively simple cell, but there's still a lot of different components to the cell, and we're going to talk a lot about that in this lecture. So the key features, the things we want to focus on with our bacterial cells, are that they are small, so they're usually less than 2 micrometers in size. So when we think of a micrometer is 1/1,000,000th of a meter. They're very, very small. A eukaryotic cell, on the other hand, is usually about 10 micrometers in size. So you can see the difference in size there. Your prokaryotic cells or bacterial cells have their DNA just floating freely in the cytoplasm. And you can see that in this figure here, that purple kind of spaghetti looking material. That is your DNA, and there's nothing separating it from the rest of the cell. Right? Our bacteria have a cell wall, and that cell wall is made of a substance called peptidoglycan. And we're going to talk a little bit about peptidoglycan here as we go through the lecture. They do not have internal membranes, all right? There's no internal compartments inside of our bacterial cells. They may have some internal structures, and sometimes you'll hear the word organelle thrown around to describe those structures. For this class, for my purposes, if it doesn't have a membrane around it, we don't call it an organelle. Because in my book, an organelle is a membrane bound structure in the cytoplasm of a cell, and prokaryotes do not have those membrane-bound structures. So when we think about bacterial cell shape, there's 3 main types of bacterial cells with regard to shape. We have these spherical shaped cells. Those are called cocci, or coccus for singular. We have our bacilli or bacillus. Those are your rod-shaped cells. And then we have our vibrios. Vibrio means kind of half moon. These are these curved shaped cells. These are going to be your most common shapes that we see in our bacteria. The shape of our cell is determined by its genetics, by its DNA, and by its cell wall structure. Here are some other examples of bacterial cell shapes. So this one up at the top, it's kind of like a really short, rounded rod. We call that a coccobacillus. We see those commonly, particularly when our rod-shaped cells are starving. We sometimes will see that where they shorten up and form this coccobacillus. Some cells just look like this all the time. This next one that you see is your spirillum. Your spirillum is this spiral-shaped cell. It's very long and flexible. And then we have the spirochetes. These tend to be more rigid in shape and more tightly coiled. And they also have this kind of corkscrew shape to them. In addition to the different shapes of our cells, we can describe our cells based on their arrangement, how those cells tend to kind of group together, and then the way that we see them underneath the microscope. So your cocci, they can occur as a single coccus, right, just a single sphere. Oftentimes they can appear as 2 cocci that are held together. That is called a diplococci. Sometimes your cocci can be held together in groups of 4. That group of 4 is called a tetrad. Sometimes our cocci will form long chains. Those are called streptococci. You've probably heard of streptococci because most of us have had some sort of encounter with strep throat, right? Strep throat is caused by a bacterium that is a streptococcus. We also have staphylococci, another one you've probably heard of. Staphylococci are cocci that kind of group together in clusters or grape-like clusters. That's where the name staphylo comes from. It means grape-like. When you view these under the microscope, they kind of look like clusters of grapes. Similarly, we have terms to describe our bacilli. So you may have a single bacillus, that would be a single rod. You may have a streptobacillus. And there are other terms that we can use to describe our groupings of prokaryotic cells. These are going to be your most common. Okay, so we are going to focus first on the cell components inside of our bacteria that are intracellular. And then we will look at our extracellular components. So I've got these two lists kind of divided up for you. Make sure when you're going through and studying the material for this chapter that you really focus and you know what each one of these structures is and what its purpose is for the cell. So the first structure that I'm going to talk about is the nucleoid. So this term, nucleoid, is one that can confuse students because we have three terms that we're going to encounter. Nucleoid is something that we see in bacterial cells and in our archaeal cells as well. And then in eukaryotic cells, we have the nucleus. And then inside the nucleus, there's something called the nucleolus. All right, so there's three terms, nucleoid, nucleus, and nucleolus. They all sound very similar, but they mean different things. So the nucleoid, remember, bacterial cells, prokaryotic cells, do not have a membrane that separates the DNA from the cytoplasm of the cell. Instead, what will happen inside the cell is the DNA kind of winds up around itself. It's called supercoiling, and it gets real concentrated in certain parts of the cytoplasm. And we can see that when we use an electron microscope. And that's what you're seeing in this image here. This is a picture of, I believe, an E. coli cell. And you can see there's this really dark staining region at the center of the cell. That is the nucleoid. So within the nucleoid region of the cell, we have the chromosome of our bacteria. So bacteria have just a single chromosome, and that chromosome is shaped in a circle. This is very different from eukaryotic cells. So eukaryotes usually have linear chromosomes where the DNA is just a continuous line. And we usually have multiple chromosomes. So humans, for example, we have 46 chromosomes. Here, our bacteria usually have just one chromosome and it's shaped in a circle. And that chromosome contains all the information necessary to make the different components of the cell. So it contains all the genetic information that makes that organism what it is. Now, sometimes bacteria have an additional piece of DNA called a plasmid. A plasmid is a small piece of DNA. It too is circular, but it usually only has a couple of genes on it. And one of the really interesting things about plasmids is that they can actually be shared between cells. That's a process called horizontal gene transfer, and we'll talk more about that as we get into bacterial genetics later in the semester. But plasmids are really important to us because sometimes they can carry genes that are involved in disease. We call those genes virulence factors. They may also carry the genes for antibiotic resistance. So you can imagine if you have a plasmid that has a virulence factor or an antibiotic resistance gene on it, you don't want that to be shared between bacteria. But unfortunately, that's what they do. And that's one of the reasons why bacteria are able to change over time and be more difficult to treat or become more virulent. Like I said, we'll talk more about horizontal gene transfer later on in the semester. The thing I want you to remember about plasmids is, again, they don't have the genes on them necessary for survival or necessary for everyday life of a bacterium, but they have genes that might give them an advantage under certain circumstances. All right, again, those genes, there's usually only a couple of them on a plasmid. The next thing I want to talk about is our ribosomes. So hopefully you remember from your Bio 1 class, ribosomes are the structures within cells that are responsible for making proteins, and they're found in the cytoplasm of both your eukaryotic cells and your prokaryotic cells. Now, DNA holds the information of a cell, right? It holds all that genetic information in it. DNA is used to make RNA, and then RNA is used to make protein. The ribosomes read messenger RNA and construct proteins based on that messenger RNA. So that's their job, is to read the mRNA. Ribosomes have two main components to them. They have a small subunit and a large subunit. Those two subunits or pieces come together to form the complete ribosome. Now you'll notice in this figure, we have a couple of numbers thrown on here. And at the end of each one of those numbers, it says S. So we have 30S, 50S, and 70S. S is for Svedberg unit. Svedberg units are used to quantify both size and shape of a molecule. We're not going to get into the technical details of how that happens, but you'll notice that our two subunits, the small subunit and the large subunit, you have the 30S and you have the 50S. When they come together, they form the 70S complete ribosome. All right? And that complete ribosome is different than what we see in our eukaryotes. So eukaryotes have what's called an 80S ribosome. That little difference is because of a difference in size and a difference in shape between the two molecules. It's really important because we can capitalize on the difference between bacterial ribosomes and eukaryotic ribosomes when we treat patients with antibiotics. Some of our antibiotics inhibit the bacterial ribosomes. They interact with bacterial ribosomes, but don't affect our own ribosomes because of the differences in the molecular shape, right? So it's those minor differences in structure that become important when we start thinking about treatments and we start thinking about drugs. All right, so remember, bacteria have a 70S ribosome, and ribosomes are responsible for making proteins. The next thing I want to talk about is inclusions. Now, inclusions is kind of a broad term. It just describes an area of concentrated material within the cytoplasm of the cell. Sometimes those are called starch granules or chromatic granules or magnetosomes. There's lots of different types of inclusions, and it just depends on the different organism what kind of inclusion it might have. The key thing about inclusions is that, again, there's no membrane around these areas of concentrated material. You may have kind of a protein shell around that material. You may have a layer of lipids around that material, but there's no phospholipid bilayer dividing the inclusion material from the rest of the cell. All right. Now, inclusions can have very specialized functions, and it just depends on the organism. But we can see them under the microscope, especially when we use the electron microscope. All right, the next structure that I want to discuss is one that's really important. I'm going to spend a little bit more time on it, and that's the endospore. Endospores are used for survival for certain organisms. So not all bacteria produce endospores. Only a few microorganisms do. One microorganism that produces endospores is one that you might be familiar with. It's called Clostridium difficile, or C. diff for short. Endospores are formed when a microorganism encounters an environment that's unfavorable. That means maybe it's starving, or it's gotten too hot or too cold, or it's starting to dry out. There's a lot of different things that can cause an organism to form endospores. But essentially what's inside of an endospore is a copy of the chromosome, some essential proteins, and then it's surrounded by a plasma membrane, and then outside of that you have a very thick coat of protein. And that thick coat of protein, called the cortex, That protects that information that's stored inside the endospore from drying, from changes in pH, from radiation, from heating, from all sorts of chemicals. These things are tough. In the case of C. diff, about the only chemical that can really effectively destroy the endospores is bleach. All right, so that's why we use things like bleach wipes in hospitals as a way to try to eliminate endospores from the environment. Because if you have a patient who has C. diff and they're in a hospital room, they may release the endospores. Now, I should say, C. diff causes, unfortunately, massive diarrhea and vomiting. It's a very unpleasant illness. And so you get endospores and bacteria released from the patient, essentially from both ends, all right? And that can contaminate the environment. Now, if it was just vegetative cells, we can clean those up pretty easily. But because there's endospores that are part of these cells, the endospores can persist in the environment for a really long time. And then the next time a patient encounters that spore, and maybe they accidentally ingest that spore, the endospore will pass through the stomach, and that change in pH that happens there can trigger the endospore to germinate. All right. So what happens is the endospores are not growing, they're inert, right? They're kind of like a dormant phase like we see with our seeds of our plants, and then they will germinate just like a plant germinates when they hit the right environment again. So this is what spore formation looks like. First, you have copying of the genome. You have the genome encased in membrane, and then it gets coated by additional membranes and the cortex proteins and additional proteins, and then eventually that spore is released from the cell. Like I said, the spore itself is not active, all right? It's what we call cryptobiotic, all right? There's no active metabolism going on inside that spore until it reaches that favorable environment again, and then it starts to act and it starts to grow. The next structure that I want to discuss is the plasma membrane. The plasma membrane is the barrier between the inside of the cell and the outside of the cell. Now, with the plasma membrane, it is composed of two parts, phospholipids and proteins. And together, the phospholipids and the proteins create a selectively permeable barrier, meaning that some materials can get in and out of the cell where others can't. They cannot easily move across that membrane. One of the reasons why materials don't easily move across the membrane is because of the phospholipids. Phospholipids form what's called a lipid bilayer, where the heads of the phospholipids that contain phosphate, they have charge to them. They're going to face out, right, towards the cytoplasm or out towards the exterior of the cell. The tails of our phospholipids face inward. So you get this kind of sandwich effect like you see here in this picture where the tails are facing one another and the heads are kind of on either side of the membrane. That phospholipid bilayer that we see here has a central component of those tails that are water-fearing. They're hydrophobic. Because of that, some materials easily move across the membrane and others don't. We say that the membrane is selectively permeable. It allows some things across, but not others. The items that can easily move across, they have a couple of main characteristics. One is that they're nonpolar. Remember, hydrophobic substances, they don't like water, they don't like anything that has charge or partial charge. In addition, for something to easily move across a membrane, it also has to be really small. So the things that we usually see that can move across those lipid bilayers are things like gases, oxygen gas, carbon dioxide, like we have pictured here. Items that do not move easily across the membrane are going to be things that have charge or are polar, right? So that sodium we see there, it's a really small ion, but because it's got that charge, it can't freely move across that lipid bilayer. Water also is polar, so it has partial charge. It does not easily move across that lipid bilayer. Other molecules that can't move across the lipid bilayer, they may not be able to because they're too big, right? So glucose, like we see in this picture, is just too large to freely move across that membrane. So for items to be able to move across the membrane, we have a couple of different ways that they can do so. We have 4 types of movement, right? We have what's called simple diffusion, we have facilitated diffusion and osmosis, and then finally we have what's called energy requiring transport. Simple diffusion, facilitated diffusion, and osmosis, those are all examples of passive transport. That means it doesn't require any input of energy. Energy requiring transport is also sometimes called active transport. So diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So what we see in this picture on the screen right here is 2 different areas, right? Where we have the red molecules that are kind of bunched together and some that are far apart, right? The area where they're bunched together, that's an area of high concentration, and when they're spaced further apart, that's low concentration. That difference across space is called a gradient. In diffusion, molecules move down a concentration gradient, right? So a gradient is just a difference across space, right? In this example, my molecules are going to freely move in the direction of the lower concentration. Right, so diffusion is due to what is just random motion of molecules, all right? Molecules are constantly in motion. Simple diffusion with a membrane, here in this picture, I have an area of high concentration of my red molecules and an area of low concentration of my red molecules. And what we're looking at, the molecules are dissolved in water. So the blue represents my water molecules. That's going to be my solvent. In this situation, my red molecules can easily diffuse across the membrane. They might be oxygen or carbon dioxide. So they just freely move directly across the membrane from the high concentration to the low concentration. So that is what we call direct diffusion of molecules across a membrane, simple diffusion. Right? They're going down their concentration gradient. Facilitated diffusion is similar, except here the molecule, my green molecule, cannot interact with those phospholipid tails, right? For whatever reason, maybe it's polar or charged. So it can't move across that membrane directly, so it has to move through a protein. So in facilitated diffusion, we have the diffusion of molecules across a membrane down their concentration gradient using a protein, and that protein could be either a channel or a carrier protein. So channel proteins, just like it sounds, they create a channel across the membrane. So they transport molecules through a narrow channel that has a specific size and shape to it. Where carrier proteins, on the other hand, they change shape as the molecule binds and allow for the molecule to move across. Again, it's down that concentration gradient. The only real difference here is that carrier proteins are much more specific about what they carry across the membrane, right? But it's still facilitated diffusion. Molecules are moving down their concentration gradient. Osmosis is the diffusion of water, all right? So again, these water molecules are going to move down their concentration gradient. So in this picture, my water molecules are blue and my solute is purple, all right? Now on my figure here, I have a protein called an aquaporin. An aquaporin is a channel that is specific for water molecules. So it allows water molecules to easily flow across the membrane. So my water can move from the area of high water concentration to the area of low water concentration. It's all relative, right? Based on the relationship across the membrane. So tonicity is a term we use to describe the concentration of water on either side of a membrane. So this is really important because people tend to get confused on this term. Tonicity describes the relative concentration of solute in two solutions, and usually those two solutions are separated by a membrane. In this example, on my side where I have lots of purple solute, I have very low water concentration relative to the other side. That side with the high concentration of solute, we say that it is hypertonic. And the side where the concentration of the solute is really low, we call that hypotonic. Those terms are relative to each other, right? Because they're separated by that membrane. We have two solutions separated by a membrane. Hypertonic and hypotonic are important terms to understand because we can use tonicity as a tool to control microbial growth. So you've probably heard of using salt to preserve foods. Well, what we do when we add salt to foods is we create a hypertonic environment and we actually draw the water out of cells. We actually remove water from cells, and that prevents microbial growth. So we've talked about our passive transport, simple diffusion, facilitated diffusion, and osmosis. Now I want to talk about our energy requiring transport, the movement of molecules against their concentration gradient. In order to do this, we're going to need some form of energy, some input of energy. This process is also called active transport. So the energy source for this transport is going to be adenosine triphosphate, the energy currency of the cell. Now remember, ATP is a high energy molecule. It has 3 phosphate groups attached to it. When we remove one of those phosphate groups, we release energy. In energy requiring transport, we use that energy to move the molecules across the membrane. So I've got an example for you. Here is an example of a membrane pump. So this is called the sodium-potassium pump. It can be found in eukaryotes and prokaryotes. A sodium-potassium pump, it moves sodium out of the cell and potassium into the cell. And you'll notice over there on the right-hand side, the sodium is high concentration outside and the potassium is high concentration inside. So we're moving molecules uphill, right? We're moving them to the area of higher concentration. In order to do that, we're going to need that input of energy from ATP. So what happens here is we're going to couple the hydrolysis of ATP to this movement by causing the change in shape of the protein pumps. So you'll see the ATP is down there on the bottom. It's inside the cell, and it binds to the sodium-potassium pump. At the same time, the pump is binding some sodium, right? And there's a chemical reaction that occurs where the phosphate is transferred from ATP to the pump itself. And in that process, we're releasing energy and the pump changes shape, right? And that change in shape releases the sodium out of the cell. It's now been moved against its gradient. Well, then you have another step that occurs where now that the pump is open to the outside and it's got this change in shape, it can bind potassium. Well, when we remove the phosphate from the pump, it changes the shape back to where we started, and the potassium is now released into the cell. So again, through the change of the shape of the protein in sequential order like this, we're able to move molecules across the membrane against their gradients. This is only possible with the use of ATP. There is another form of active transport that occurs in bacteria that we don't see in eukaryotes, and that's called group translocation. And group translocation, as molecules are being moved into the cell, they're actually chemically altered, they're changed, they're phosphorylated. And that's a unique transport pathway that we see in bacteria that we don't necessarily see in our eukaryotic cells. Again, that's called group translocation. Okay, so we're going to move out of the cell and into the external environment. And the first thing we're going to encounter is the cell wall. The cell wall is exterior to the plasma membrane. So the cell wall is composed of a material called peptidoglycan. It gets that name because it is composed of both sugars and peptides. Right? So peptides are short chains of amino acids, and then glycan stands for sugar. In the case of your peptidoglycan, the sugars that we find are N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM for short. And in this structure, they occur in this kind of repeating pattern where you have NAG, then NAM, then NAG, then NAM. So they come in these long chain polysaccharides. Attached to those sugar chains, we have short chains of amino acids or peptides. Now, we have two different types of cell walls. We have gram-positive cell walls and we have gram-negative cell walls. And you can see in the diagram below that the orientations and arrangements of the peptides are a little bit different between the two types of cell wall. So the job of the cell wall is not to control what goes in and out of the cell. That job is left to the membrane. The job of the cell wall is to protect the cell from harsh environments. It gives it structure, it gives it support. It determines the shape of the cell. Right? But remember, it doesn't determine what comes in and out of the cell. It's this kind of loose mesh network. So there's lots of holes in the peptidoglycan or spaces in the peptidoglycan. So like I said, there are two types of cell wall. There is the gram-positive cell wall and the gram-negative cell wall. The gram-positive cell wall, the main feature of it is that it has a very thick layer of peptidoglycan. So you can see that in the figure on the left. That blue is the peptidoglycan layer, and you just see one layer over the next, over the next, over the next, all right? So, and again, it's sitting right above the plasma membrane. In addition, the gram-positive cell has a substance called teichoic acid embedded in the peptidoglycan layer. That is characteristic of our gram-positive bacteria, and we don't see it in our gram-negative bacteria. On the other side here, on the right, we have a picture of a gram-negative cell. And the gram-negative cell is different because it has a very thin layer of peptidoglycan, and then it has a second membrane or outer membrane that extends outside of the cell, outside the cell wall. Right, kind of like a sandwich. So you have this peptidoglycan layer that's sandwiched between two membranes. And that space between the two membranes is called the periplasmic space. So you can imagine transport into a gram-negative cell is going to be a little bit more complicated than it is for a gram-positive cell, because not only do you have one membrane, but you have two membranes to get across. And we're going to talk a little bit more about this outer membrane here in a second. So we have two main types of cell wall. You have a gram-positive cell wall, you have a gram-negative cell wall. They get these names based on how the cells appear using something called the Gram stain. So the Gram stain is a process where we stain bacteria so we can visualize them under the microscope. And we learned about differential stains in our last chapter, right? Differential stains stain bacteria differently depending on their characteristics. So those bacteria that have that thick peptidoglycan layer, they stain purple, which we call gram-positive, and the ones that are have the outer membrane on them, they stain pink, so we call that gram-negative. And you can see in this image here, I have both gram-positive and gram-negative cells both in this smear preparation. Although it's a little bit blurry, you can see that the gram-positive cells are cocci, right? They're those round-shaped cells, and the gram-negative cells are actually rods. They're bacilli. Okay, so let's talk a little bit more about those gram-negative cells because they are unique in that they have this outer membrane. The outer membrane is not composed of the same phospholipids that we see in the plasma membrane. The outer leaflet, or the outer layer of lipids, is a substance called lipopolysaccharide. So you have a lipid that is embedded in the membrane, and that lipid is called lipid A. And then lipid A is attached to a polysaccharide. That's why it's called lipopolysaccharide. And that polysaccharide extends out from the gram-negative cell surface. Lipopolysaccharide, or LPS for short, is also called endotoxin. So when this substance is in our bodies, in the human body, it is highly inflammatory. And our body reacts negatively to it. So it can actually, if it's in high enough concentration, it can lead to shock and even death. And that's because of the presence of this lipid A and these polysaccharides that make up the lipopolysaccharide. Like I said, it's also called endotoxin. Endo because it's part of the cell. It's not an actual toxin that's released by the cell, but it's actually part of the cell. It's embedded in that outer membrane. And we'll talk more about this as we get on later on in the semester when we talk about pathogenicity of microorganisms. So again, gram-negative cells, they have a standard plasma membrane. Then they have a periplasmic space that contains the peptidoglycan cell wall. Then they have this outer membrane, and that outer membrane contains lipopolysaccharide, or LPS, or endotoxin for short. All right, we do also have some bacteria that have very unique cell walls. One particularly unique type of cell wall is our mycoplasma. Well, they're unique in the sense that they don't have a cell wall. So these are bacteria that actually don't have any sort of cell wall structure. But they have some chemicals that are included in their plasma membrane to help stabilize the cells. The other unique type of cell wall is our acid-fast bacteria. Acid-fast bacteria contain a waxy substance called mycolic acid. So you may have heard of Mycobacterium tuberculosis or Mycobacterium leprae. Those are a couple of famous pathogens that are acid-fast bacteria. They're known as acid-fast positive, meaning when we stain them using the acid-fast stain process, they appear this kind of purpley fuchsia color that you can see in the image below. So acid-fast bacteria, they're actually considered gram-positive because they have that thick peptidoglycan cell wall, but then exterior to that peptidoglycan cell wall, they have mycolic acids. Mycolic acids are very waxy, and that prevents the cells from drying out, and it also protects the cells, particularly when they're taken in by our cells of our immune system. So it protects them from being digested by our cells. So it's an important component of those organisms' pathogenicity. All right. So again, acid-fast bacteria are bacteria that contain mycolic acid in their cell walls. Okay, so now we're going to move beyond the cell wall and talk about some structures that we find external to that cell wall. This includes the extracellular coatings, that's the glycocalyx and the S layer, and our extracellular appendages. So this includes our flagella, our fimbriae, and our pili. The glycocalyx is composed of carbohydrates. Glyc is for sugar. It's a matrix of carbohydrates that we find external to the cell wall. So you can see in the figure below that you have that kind of bluish green coating. That is, in this case, that's a capsule, but capsule is a type of glycocalyx. So it's outside, it's beyond the cell wall, and it is made up of carbohydrates. On the right side of your screen, you have an example of what's called a capsule stain. These carbohydrates, they're very difficult to stain. The different types of dyes that we use don't tend to stick to our carbohydrates of our glycocalyx, so we oftentimes will use negative staining in order to visualize them. So that white halo that we see around the cells in that staining, that represents the glycocalyx. In this instance, of course, it's a capsule. So there are two types of glycocalyx. There's the capsule that I've already mentioned. The capsule is organized and it's firmly attached to cells. And then we have the slime layer, which is loosely attached and less organized than the capsule. Now, the functions of our glycocalyx primarily allow for the cells to attach or adhere to surfaces and to other cells. It also gives a certain amount of protection to the cell. For example, a cell that has a slime layer may be part of a biofilm. And in a biofilm, you have these communities of cells that are all kind of stuck together. They have all this extracellular matrix that surrounds them, and it's very difficult for disinfectants and other substances, even antibiotics, to penetrate into the biofilm. Part of that is due to those slime layers in that glycocalyx. Another example where a glycocalyx can protect cells is in the case of a capsule. So cells that have a capsule can hide from the human immune system. So it protects them from being eliminated by our phagocytic cells, those cells that are bacteria eaters. So glycocalyx actually can have a very important role in virulence, because strains, for example, of Streptococcus pneumoniae that have a capsule are virulent. They can cause disease, but strains of that same organism that do not have a capsule, that do not have a glycocalyx, don't cause disease at all. So that's just an example of where the glycocalyx can not only protect the cell, but also play a role in the disease that an organism might cause. So we call that pathogenicity. All right, similar to a glycocalyx, so the glycocalyx is a sugar coating or a carbohydrate coating on the outside of a cell. The S layer is a coating of protein on the outside of a cell. Now, these proteins usually are found in very regular patterns, like you can see in this picture. It almost looks like a little carpet on the surface of this cell. S layer proteins can be found in both bacteria and archaea. And here, the function of them is not fully understood. We're still kind of studying and trying to understand the role of S-layer proteins in protecting cells. We do think that they're involved in adherence. Okay, so we've talked about the extracellular coatings. Now we need to talk about our extracellular appendages. So the first one I'm going to talk about is the one you probably have heard of before. That's our flagella. Now, flagella are these long filamentous appendages that are made of a protein called flagellin. So they look like a long tail that extends off the surface of the cell. That's the external component of the flagella. But the flagella actually start inside the cell. So they have this structure called a motor, and it has a rotor, just like your wheels on your car have an access to them. You have a rotor rod that sticks down into this motor and actually connects to that filament we see sticking outside of the cell. So that allows for the cell to move this flagella according to different stimuli, different things, signals that a bacterium might be following. So the structure is a little bit different between gram-positive and gram-negative cells because in a gram-negative cell you have that outer membrane. So in a gram-positive cell you just have one kind of motor arrangement. In a gram-negative cell you have the motor and then you have a set of rings that embed this flagella into the outer membrane. The function of the flagella is to allow the bacteria to move, right? Their job is to propel a organism through an aqueous environment, through a watery environment. Now, some bacteria have just one flagella. We call them monotrichous. Sometimes they have flagella on either end. We call that amphitrichous. Sometimes they have a tuft of flagella on one end. That is lophotrichous. And then sometimes they're just covered entirely with flagella, and we call that peritrichous. So there's different arrangements that we have of flagella, and it just depends on the organism how many flagella they may have. The way that the flagella moves, it's got that motor protein that's embedded into the cell. That motor protein turns the flagella in such a way that it rotates kind of like a boat motor or a boat propeller. So you can move that flagella clockwise or counterclockwise. And by moving the flagella one direction or the other, that allows for different types of movement. So counterclockwise movement, for example, allows the flagella to move kind of in a straight line. But if you reverse the motion of that flagella, that might cause the bacterium to tumble or roll. So you've got kind of this different patterns of movement that you see in our microorganisms based on which direction they're moving the flagella at any given time. So this is an example of a bacterium that is moving in response to something. In this case, it's a chemical that it's attracted to. So it starts moving in one direction using those flagella to propel it forward. That's what we call a run. And then it reverses the movement of the flagella, and that is a tumble. It's kind of like if you've ever gone canoeing or paddling, you know, if you backwards paddle, it helps turn the boat. It's very similar to that with your flagella. They're rotating their flagella the opposite direction so that they can kind of change directions. And then they can, from there, move in another direction. So the flagella allow the bacteria to move towards something or sometimes away from something. All right, so that's flagella. They're used for movement. The next external appendage I want to talk about is fimbriae. Fimbriae are much shorter than flagella. All right, so I've got this picture here for you, and you can see this is an E. coli cell. And you see these very thin fibers that are kind of extending off of the cell. These are kind of short hair-like structures. You don't have a flagella to compare it to, but they're much shorter than the flagella. These are called fimbriae. Fimbriae are used primarily for attachment. They're protein polymers, and they attach cells to surfaces. For example, E. coli, some strains of E. coli use fimbriae to attach to the inner surface of the bladder and the urinary tract, and that's why they're able to cause urinary tract infections. The last appendage is our pili. Pili, and this picture actually gives you a good perspective. If you look at that bacterium up on the top left of the picture, you can see some fimbriae extending off of the cell. They kind of look like short little hairs. Those are the fimbriae, and then we see this pili. Pili are much larger, and they are composed of a protein called pilin. And the pili are used for a couple of different purposes. They are long and filamentous, so they're very long compared to our fimbriae, but they don't have that motor structure that we see in our flagella. Instead, these are used primarily for attachment to surfaces and attachment to other cells. One way that bacteria use pili is through a process called conjugation. So in conjugation, remember how I talked about how plasmids can be shared from one cell to another? Conjugation is one way for bacteria to share genetic information. So essentially what happens is the pili is used to attach to a neighboring cell, and then it actually acts like a little connection between the cells, allowing for the transfer of that DNA from one cell to another. Now that we've covered all of the different components of our bacterial cells, what I want to do is take a few moments to talk about archaea, those other prokaryotes, right? Archaea do not have a nucleus, so in that way they're similar to bacteria, but it turns out that they're just a little bit different, okay? And in fact, they're actually more closely related to eukaryotes than they are to bacteria. And we base that on the genetic similarity of these organisms to eukaryotes. When we look at the DNA sequences, they align more with our eukaryotes. So archaea are different genetically than bacteria. They also do not have a peptidoglycan cell wall. So some of them don't even have a cell wall, and others have cell walls that are composed of other materials. Sometimes we call that cell wall material pseudomurine, which is a way of saying that it's like peptidoglycan, but it's not peptidoglycan. So that's a big distinction between archaea and bacteria, is that difference in the cell wall components. Another important thing about archaea is that they're extremophiles. And I know I mentioned this back in my first set of lectures, that archaea tend to live in very extreme environments, things like hot springs or salt lakes, deep sea vents. We see archaea in those locations, but they're not restricted to living in extreme environments. So some archaea even live in our bodies, in our gastrointestinal system, or on our skin. So they have a unique metabolism, so they tend to use biochemical reactions that are different than what we see in our bacteria. For example, some of our archaea can take carbon dioxide and convert it to methane gas. That's something that we don't see in other organisms. So that's an example of a unique metabolism in archaea. Some are actually, like I said, some actually live in the human body and are part of the human microbiota, those organisms that live in and on our body. And we're still learning about these organisms. They're very difficult to grow in the laboratory. So, and as far as we know, they don't cause any human disease. So largely they've been ignored by scientists over the years. So this is something that we need to study more about. What is the role of archaea in human health? Is there a role for archaea in human health? So like I said, there's no known human pathogens, no disease-causing archaea. So this is really all I wanted to say about archaea. Like I said, we're not going to study them much this semester. We're really going to be focusing primarily on bacteria and other pathogenic microorganisms. So with that, I wanted to talk a little bit about how we classify our prokaryotes. You know, initially we grouped bacteria and archaea together because our original tools for classifying living things were very rudimentary. Think about how we first discovered microorganisms back in the 1800s. It was by using our microscopes and studying the organisms under the microscope. We started to do some staining techniques and growing microbes in the laboratory. Scientists like to group organisms to kind of make sense of the world. And we put all the cocci together in one group and all the bacilli in another group. And then we started to divide those up based on gram-positive and gram-negative. Where did we find those organisms? And that started to help us make sense of the diversity and the different types of bacteria and archaea that were out there. But we have lots of other tools in our arsenal now that help us classify prokaryotes. So as our science advanced, we started to use what's called biochemical classification, where we started to ask, well, what chemical reactions can an organism perform? Does it ferment a particular kind of sugar? Or can it detoxify a kind of chemical? So there's lots of different biochemical tools that we use to determine the type of microorganism that we're studying. And we're going to learn about some of those as we go through our lab activities. Of course, nowadays we have some even more important tools, our genetic tools, our molecular tools that allow us to compare DNA sequences of our microorganisms. And by looking at the sequence alignment, how similar or how different the DNA of two organisms is, we can start to decide how related they are to one another. So this first started by looking at just what's called ribosomal RNA sequencing. And we initially developed our three-domain system of bacteria, archaea, and eukarya based on this ribosomal RNA sequencing. It was a scientist by the name of Carl Woese who proposed this system for us. But then now we have even more ability to sequence DNA, and we can do what's called whole genome sequencing, where we can find out the genome of an entire organism within a matter of hours. So our computing technology and our laboratory technology has advanced us to that point where we can really fine-tune how we classify our microorganisms. So there's a book called Berge's Manual that was used as like the definitive guide for identifying microorganisms. If you weren't sure what kind of bacteria you had, you could use Berge's Manual to figure that out. So it is what we call the definitive guide to bacterial classification. Now, as our science has advanced, we now have two Berge's manuals. We have Berge's Manual of Systematic Bacteriology, and we have Berge's Manual of Determinative Bacteriology. The Systematic Bacteriology Manual is focused on evolutionary relationships between different microorganisms, where determinative bacteriology is more focused on the physical characteristics of our microorganisms, and this can be more important particularly when we are working in a clinical laboratory, right? So as you can imagine, there's a lot of confusion in the world of bacterial classification. And that has a lot to do with the fact that it's very difficult to define what we mean by a bacterial species. When we think about larger organisms, about eukaryotes, particularly sexually reproducing organisms, we define a species by whether or not those organisms within that species can mate with each other and reproduce, right? But we can't do that with bacteria, right? We also can't really study their behavior real extensively, I mean to a certain degree, but you know, they're microscopic, they're difficult to see. So bacterial species definitions are really kind of loosey-goosey, and it's, pardon my pun, but it's constantly evolving. We are still trying to figure out the best way to define a bacterial species. But for now, we say that it's a collection of bacterial cells that share common traits. All right, so sometimes we divide those species down into further groups, right? So we might have subspecies or strains, or even serotypes, which is a way of classifying organisms based on how they react with antibodies. So I'm going to give you an example of what I mean here. So E. coli is a species of bacteria. You probably have heard of it many times. I know I've mentioned it many times already today. E. coli is a rod-shaped microorganism. It is gram-negative, so it has that gram-negative cell wall. And it does not make endospores. So those are three important physical characteristics of this organism. In addition, it can live with oxygen or without it. So that's another tool we can use for grouping and classifying organisms, is whether they are aerobes or anaerobes. Okay. And then in this case, E. coli is commonly isolated from the habitat of the gut. It's really easy to grow in the lab, and it grows at body temperature. And then finally, we can use some biochemical tools like looking at sugar fermentation to say, okay, well, if it has all of these characteristics and it also ferments lactose, it's probably E. coli. Now, with that said, there are hundreds of strains of E. coli. So even though we've defined this species of E. coli, we've got this list of characteristics that makes it E. coli, there are so many different strains of E. coli, and frankly, there's a lot of genetic diversity within this single species. So you'll notice sometimes as we're going through the semester that bacterial names can be changed or shuffled around over the last several years as we've started to move towards more genetic tools. We have even reclassified microorganisms. So there can be some confusion in this area. And this is also why I don't focus a lot of my time and attention on making sure that you know which classification a microorganism falls into, other than being able to tell me that it's a bacterium and it's gram-positive, gram-negative rod, or cocci in this particular class. Okay, so that concludes the lecture on prokaryotic cells. We will continue in our next lecture talking about our eukaryotic cells and how they're different from prokaryotes and some of the different types of pathogens that we see in our eukaryotes.